Good afternoon. Okay, before we start, I hope you can browse our guidelines for this webinar. Maybe I'll give you a minute for our guidelines. Okay, so hello, good afternoon. Um, I would like.
Hi, I was just about to offer an afternoon pick me up.
Okay, wait
Ask
lang.
me for a joke.
So before we start, I would like to thank Sir Richmond Lim and Sir Christian Evasco for making this webinar possible online and, of course, live on Facebook. Good afternoon to all our faculty and FEU students. Good afternoon also to our participants from the Department of Education, from Mindanao State University, from the Philippine Science High School, from University of the Philippines, and from Ateneo de Manila University. Thank you so much for attending our webinar. I would also like to acknowledge the presence of our FEU officials from the Institute of Arts and Sciences, Dean Rowena Capulong Reyes, our Associate Dean, Sir Mark Isla, and of course, our dearest Department of Mathematics Department Chair, Professor Frederick Helia. Again, good afternoon, and welcome to our webinar entitled Making Sense on the Physical World, One Image at a Time. Ayan. I am Miss Isa Bernaldez, and together with Sir Alvin Cola and Ma'am Hernal, we will serve as your moderators for today. In this pandemic has put us into a very challenging but interesting circumstances in terms of teaching and learning. That's why we will dedicate this webinar to our teachers, to our dear students, and for researchers. This webinar wishes to provide ideas and practical approach, especially in conducting laboratory activities in physics. Now, it will specifically discuss image processing and analysis program that may be used and applied to research, specifically in data science, biology, material science, and even in agriculture. So to start, to give us an opening remarks, I would like to request our department chair, Sir Professor Frederick Helia, to have the remarks. Sir?
Okay. Our guest speaker, Dr. Merjibel Tingal, Institute of Arts and Sciences Dean, Dr. Rowena Capulong Reyes, IAS Associate Dean, Mr. Mark Salvador Isla, faculty members, deputy teachers, Philippine Science High School teachers, ladies and gentlemen, good afternoon. It is my pleasure to welcome everyone to our physics webinar series titled Making Sense of the Physical World, One Image at a Time, using ImageJ in work from home science experiments as part of our IAS virtual activities supporting education, or IAS Vase, founded by our Dean, Associate Dean, and some program heads. This webinar aims to provide an introduction on the use of ImageJ in scientific image analysis. ImageJ, by the way, is an open source Java-based image processing and analysis program that is extensively used in the biological and physical sciences. With its additional plugins, ImageJ has contributed a lot to the world of visual programming, data representation, tool blending, prototyping, automation, and scaling and reproducibility. I hope you will enjoy the rest of our virtual conference, and thank you.
Thank you, Sir Helia, for that welcome and some overview of our webinar today. And now, to introduce to us our speaker, may I call on my dear colleague, Ma'am Mary Joyce Hernal, to introduce Miss Dengal.
Hi.
Ma'am Joyce?
Hello. Thank you, Mama Icar. So, good afternoon. Good afternoon, everyone. So, I am honored to be with you this afternoon and to be given the opportunity to introduce our guest speaker. Our speaker is an associate professor from the Mindanao State University main campus who specializes in complex systems, granular matter, active systems, and video image processing. In fact, she introduced yours truly to the fascinating world of ImageJ and image analysis. She graduated cum laude from the Mindanao State University main campus in Marawi City with a bachelor's degree in physics. Years after, as a DOST scholar, she earned her master's degree in physics from Mindanao State University, Iligan Institute of Technology in Iligan City. By 2019, she received her doctorate degree in physics from the Ateneo de Manila University through the DOST SEI Scholarship Grant. Aside from being an educator, a mentor, and an image processing enthusiast, our speaker also enjoys arts and crafts. My Athenian classmates and I call her the Jill of all trades because she has a wide span of skills from programming to computational physics to arts to dressmaking and so many others. With all her accomplishments and talents, an admirable facet of our speaker is her constant humility. So without further ado, please join me in welcoming our speaker, Dr. Mergibel D. Dengal.
Thank you very much, Miss Joyce. Miss you. So Joyce and I, we go back several years na, no? So colleagues kami sa Ateneo together with Sir Al. So thank you very much, Ma'am Joyce. So allow me to share my screen with you today. So thank you to everyone who's here and who are still joining. Maraming salamat po. And of course, to the FEU community for inviting me to be your speaker in today's webinar. So you might have already seen the title of this presentation or this lecture. Medyo madrama siya, no? So, making sense of the physical world one image at a time, using ImageJ in work from home science experiments. So, yung title na to, this is inspired by mga sinusulat nila about the purpose of physics, no? So, mga books, it's about making sense of the world, no? Kaya siya physics. So, yun ang naisip kong i-title. And this time, one image at a time, because we are going to learn techniques, no, in image analysis. So, if a picture paints 1,000 words, as a popular song goes, how do we extract these words and make sense of the given picture? Now, if the picture is taken from a science experiment, what tools can we use to extract useful scientific information from such picture? So in today's talk, hopefully I will be able to impart to you a certain tool or it's a very powerful tool in image analysis, and that would be ImageJ. And to do that, since ImageJ is a very vast, very vast ang kanyang applications, we will only focus on 3 introductory experiments where we can apply ImageJ. So our first experiment would be freefall motion of 2 non-identical balls. So here, makikita natin paano yung manual versus automated tracking. Second experiment is automated particle analysis of non-homogeneous granular mixtures. And then the experiment 3 would be contact angle measurements, hydrophobic versus hydrophilic surfaces. And then finally, we will wrap this up with a summary. So I hope you will join me in exploring the world of ImageJ. So let us start with a question, what is ImageJ? So Sir Frederick kanina, so meron na siyang na-introduce about ImageJ. Now, if you want to learn more about ImageJ, of course, Google is our friend. So you can type in ImageJ in your Google search bar. And the first link that you will see would be the website of ImageJ. So imagej.nih.gov. And you see that ImageJ is a Java-based image processing program. So initially, this was developed by the National Institutes of Health. So yung pinaka-application niya before was in medical, in biology and in medicine. So hindi pa siya talagang ginawa for physics. So more on bio siya before. But now the applications are parang limitless, no? If you go to the ImageJ website, so this is their old website wherein you can read about ImageJ. And if you click on features, yung pinaka-magandang feature niya is that it runs everywhere. So dahil written siya in Java, the program, it can actually run in Linux, Mac, OS X if you have your Mac computers, and then it can run in Windows, both in 32-bit and 64-bit modes. So I started using ImageJ when I was still in undergraduate. So 2010, yung first ko na paper using ImageJ. And since then, it has been my constant companion sa ano sa world of science, sa research. It's a very, very helpful tool. So if you click on download, you will see the different bundles of ImageJ that you can download for your specific machine. Now, ImageJ is actually migrating to a newer site. So they have this new site, imagej.net/imagej. And if you click on the downloads here, you will see this first FEU. So this is still ImageJ, and this is what I use. No? So it is an image processing package. It's a batteries included distribution of ImageJ, where you will find most of the scientific analysis is already here. Because if you download the original ImageJ, for example, kailangan nyo pang, wait, somebody's writing in the screen. Okay, so kailangan nyo pang kailangan nyo pa siyang i-update yung plugins to the specific plugins na kailangan nyo. But if you download FEU, or maybe if your applications are more on Bio, so you can download Bio 7, hindi nyo na kailangang i-update kasi karamihan ng plugins na kailangan nyo would already be there. So FEU is what I use. Okay. So this is how it looks like. So suppose in-install nyo na yung inyong FEU or ImageJ. So ito yung lalabas, no? And then yung pinaka-importante before starting ImageJ is to check kung ano yung memory. So since this is a video image processing program, kailangan niya ng malaking memory. But it can run on 2 gig lang naman. So kailangan niyo lang i-maximize yung memory sa computer niyo. So what you do first, you click edit, options, and then you go to memory and threads. And then in my case, 4 gig yung RAM ng PC ko. So what I use is I set 3,500 MB for that sa ImageJ alone, no? So to maximize. And then when I use my ImageJ, hindi ako nag-open ng iba pang tawag niyan, mga ibang software applications, sorry. So like I do not play videos or say especially like Photoshop for example, hindi na ako nag-open. Para lang ma-maximize at hindi ako magka-blue screen, no? We don't want that. Now, how do you know kung ano yung installed memory sa computer niyo? Well, you can just click on your PC and then say click properties. So right click on your PC, click properties, and you will see in my case I have 4 gig and only 3.7 gig is usable. So I set about 3.4 of that for ImageJ. So if you want to learn more about ImageJ and its history, there's actually a paper published in Nature Methods. So this one is published in 2012. So today, it's now 2020. So hindi na siya 25 years, no? So it's more than that. But this one traces the history of ImageJ. So this would be a good reading. But for now, let us leave the specifics of ImageJ and let us get to know it by looking at a specific experiment. So the first experiment that we are going to see is a freefall motion of 2 non-identical balls. And here we will learn how to do manual versus automated tracking in ImageJ. So the objectives of this experiment is first to observe and record the motion of 2 non-identical balls that are simultaneously dropped from the same height. So I think a lot of you who have already enrolled in physics classes, you know that this is one of the first experiments in your physics lab, right? The free fall experiment. Now, of course, in the lab, you would have your, it could be your cell phone or stopwatch to record yung stop time and yung start time ng ball when you drop it. Or you can use more sophisticated equipment. Now, in our case, we will also want to construct a position versus time graph and a velocity versus time graph for each of the 2 drop balls. And then compare the experimental value for the average acceleration of each ball to the accepted value for free fall acceleration g. So if your school is lucky, you might have had encountered this free fall apparatus, for example, from Pasco, and I'm sure a lot of manufacturers also have quite similar apparatus, no? So this one is very sophisticated. When you drop the ball, meron siyang release mechanism na mati-trigger. So that would indicate the start of your free fall motion. And when your ball drops here, magkakaroon din siya ng switch na mako-close. So you will see the time. But of course, especially now that we are in a work from home situation, not everyone could get this equipment. So how do we get our students to appreciate free fall motion, for example, or to see kung paano nila madidetermine through experiment yung value ng ating gravitational acceleration G. So this is where data analysis comes in. So for example, in this slide, we have two balls. So meron tayong maliit na marble, and then you have this bigger ball. So these two balls are non-identical. But we know that according to Galileo, these two balls would fall at the same time if we neglect the effects of friction or air resistance. No? In this case, ball 1 is solid, while ball 2 is hollow. So wala akong timbangan sa bahay. So, but I think the marble would be heavy, a little bit heavier than this plastic ball. But ball 1 is smaller in size compared to ball 2. So I have an electronic caliper at home, so I was able to measure this one. So 16 millimeter ang kanyang diameter, and 29 millimeters ang diameter ng bigger ball. But of course, we know that in free fall experiments, hindi nagmamatter yung diameter ng ating balls, right? So in physics, we consider this as point particles lang in our experiments. So let's see if those assumptions are valid no? When we get our experimental values for G. So this is a sample video of the experiment. So this is a recorded video. And I want you to take note of this part here. So meron tayong scale. So when we do video image processing, kailangan natin ng scale always. Every time we take pictures, dapat may ruler na katabi because that would be our basis for our measurements later on. Kasi di ba yung picture natin, pixels yun, di ba? So paano natin malalaman kung ilang number of pixels yung ganitong length? So kailangan natin ng ruler natin or any type of measuring tool for that matter. Pwede rin naman something na alam mo yung size, hindi necessary na ruler. Okay? So that's just the video. And then what to do with the video? Now that you have experimented and then you have recorded it in video, anong pwede mong gawin? Paano mo malalaman yung value ng gravitational acceleration G? So in this case, we have these two balls. And this one is captured at 60 frames per second using a Fujifilm X-T1 camera. Now actually, maraming smartphones ngayon na mas high power pa kaysa dito. No? So it could capture slow motion, so mas marami pang frames per second. Pero kahit naman nasa 30 frames per second, I think doable pa rin ang experiment na to. Now, if we look at our ImageJ, you will see na if you click File, Import, yung mga options lang natin, we have image sequence, raw, LUT, so this one. And sa videos, yung option lang natin is yung AVI file. Now, if you have tried recording videos in your phone, ano ba yung usual ng mga formats? Usually MP4, right? Pag ano naman, pag camera, digicam, usually .mov. No? In my case, .mov yung format. So, paano natin ngayon ilalagay ito sa ImageJ kung ang ating video ay .mov na format? So, we have two options. The first is to convert the video into .avi. So, meron ding mga freewares to do this. The second option, which is mas ginagamit ko siya, this would be convert to image sequence. So, why do I prefer this option? Kasi yung option na to, di ba usually pag magre-record ka sa bahay at mag-isa ka lang, i-click mo on, tatakbo ka dun sa setup mo, tapos tsaka ka maghuhulog, tapos babalik ka, i-off mo yung camera mo, for example, na nilagay mo sa tripod. So meron kang mga several seconds na hindi mo siya kailangan. Now, kung ilo-load mo yung lahat sa ImageJ as a sequence or as an AVI file, sa i-read niya yung lahat, mo-overwhelm yung ImageJ mo sa dami ng images. However, if i-convert mo muna siya sa image sequence, pwede mo munang i-browse sa folder, tingnan mo lahat ng hindi kailangan before and after. So, iiwan mo lang yung kailangan mo dun sa motion mismo. So, mas konti lang yung number of pictures ngayon na kakailanganin mo. And alam mo na yung first photo mo, yun na yung at time equals 0 mo, right before your motion happens. So, mas maganda siya, mas magaan. Kumbaga. So, this is what I prefer. And how to do that? Yung ginagamit ko since 2010. So, it's actually a freeware lang din siya, no? So, it's a free studio. And what you do here is you just click on DVD video. So, meron tayong free video to JPEG. Ida-drag mo lang yung AVI. And what I usually do is every frame to maximize yung frame rate natin. And then close. And then now you have a folder. At yung folder na to, ginawa niya ng 1 image per frame. So if you have 30 frames per second, so bawat second, magiging 30 images. So yun na yung ilo-load ko mamaya sa ImageJ. So this is how we do it now. So I click File. Yeah, sorry. So I click File, Import, and then Image Sequence. So I only have to select the first image. So I will zoom in. The very, very first thing that you do when you analyze an image in ImageJ is we zoom in and select a scale. So I use the straight line to draw a straight line. So when you use your mouse, you can actually hold the Shift key para straight yung maging line. And then I will click Analyze and then Set Scale. Automatically, ilalagay niya yung length ng line. So ilalagay ko yung known distance na alam ko na 25.4 millimeters yung aking 1 inch. So now I can zoom out the picture. And you will see here, so sa part na to, you will see na kanina, pixels pa dito. Pero ngayon, yung image mo, in terms na siya of millimeters. So everything now that you will measure from here would be in terms of millimeters. So pwede na tayong makagamit ng equations later on. Kasi pag pixels, hindi natin alam. Wala tayong guide. So this is how we do the manual tracking. We will use the rectangle na icon. So that's the rectangle tool, and we will draw a rectangle or a square dun sa ating object, no. Yung pinaka-importante natin dito would be the center of mass or the centroid, and you can actually check everything anyway. Pwede niyo namang piliin kung ano lang yung gagamitin niyo na data later on. But for this part, we want yung center of mass dito or the centroid. Pwedeng either sa dalawa. So i-explain natin later ano yung difference. So I have it here. Tapos, ang next mong gagawin, i-click mo lang next, yung next na figure. You will see, by the way, dito, so stop ko muna siya. So, if we zoom in here, 2/20, so you will see na yung video ko is 20 images lang. So, lahat na yun, parang ilalagay siya ng ImageJ into one set, para lang nasa ilalim yung, it's called a stack. No? So, if I'm sa baba, it will actually show yung next na figure. So, para lang yung slider na pwede mong i-slide. So, I click next. Tapos, balik ako dun sa rectangle tool. I-move ko yung aking, rectangle. Tapos, click ako ng Analyze, Set Measurements. Or, you can simply click Control-M. So, ganyan. So, you just do that. And you will think na, nako, nakakapagod kasi 20 images. But if you get the hang of it, it will come easily sa inyo. So, click mo lang, tapos next. So, Control-M, and then you do that until the whole 20 images is analyzed. So, meron ka na dito sa results. So, later on, you can just save this, and this can be imported to Excel. So, click ka lang ng click. So, you will see na lumalaki yung nag-blur siya, di ba? Because of the change in velocity. And since yung ating camera, hindi naman talaga siya yung super fast na camera na ginagamit sa mga science shows, no? Yung mga 1,000 frames per second. So si Miss Joyce, meron siyang camera, 1,000 frames per second. So pag ganun ang camera mo, hindi mo masyado makikita to, bilog-bilog talaga. But in my case, nag-o-oblong siya. So kinukuha lang natin dito yung center part. It could be the center of mass or the centroid. No? So ayan, ginagawa ko lang siya for all the 20 images. So kung iisipin mo, i-compare mo to siya sa usual mo na ginagawa sa lab, for example, in a free fall experiment. Di ba usually, pag wala masyadong equipment, yung gagawin mo is may magda-drop ng ball, tapos may mag-click ng stopwatch, tapos start and then end. And then you will do several trials of that, di ba? Pero yung makukuha mo lang is the start and the end time. Hindi mo makukuha yung yung path or trajectory ng bola, no? As you go through it. So now you just have to bumalik ba ito siya? So just do it for all the balls. And after that, pag nakuha mo na lahat, you can then save it as an Excel file. No? So I think nandito sa last. Yan, click File, Save As, and then lalabas doon is a results file na pwede nyo i-export sa Excel. Okay? So now you can see that with just one video, you have a total of 20 na data points. And bawat 20 na yan, you have your position and time. And from the position, you can get your velocity, di ba? So parang easier siya compared dun sa paghulog-hulog mo na ilang beses kang magkiklik ng stopwatch, no? So I would also like to clarify dito sa ImageJ, because you might wonder later on. So balik ko lang dito, you will see from here na yung ating measurements is that as we go down, yung ating Y is lumalaki yung value habang pababa yung bola. Okay? So this goes to show, or this tells us that yung sa ImageJ, by default, yung kanyang is if you go to the right, if you move to the right, you will see the coordinates here at the top. So it's moving, it's going more positive, and if you go downward, it's becoming more positive as well. So that means what this tells us is that the coordinate is here. Nandito yung kanyang di ba? Yung kanyang 0, 0 origin. So you can actually click invert Y coordinates in ImageJ if you want to transfer the origin. So if we do that, dun sa set measurements, invert Y coordinates, you will now see if we move to the right, ganun pa rin naman, increasing ang X. But if we move to the pababa, bababa na yung Y natin. So that means na transfer na natin yung ating Y coordinate. So as a summary, by default, ImageJ would do this. This would be our 0, 0. If you click on invert Y coordinates, ito na yung magiging 0, 0 natin. Okay? But you don't have to worry kasi minsan, tapos ka na mag-record, tapos ka na kumuha ng data mo. Kaso, pala, hindi ko na-invert. Mas gusto ko ganito. Well, pwede mo namang i-process yun later sa Excel pag alam mo naman yung size ng image mo, which is nandito. So, post-processing na lang. So, that would work either way. So, ito na yung data natin na in-import ko sa Excel. In this case, naka-invert na kasi yung Y coordinates dito, no? So, dun sa next ko na analysis. So, in-invert ko na yung Y coordinates, and then you have the X and Y na column. Ito siya actually yung ating centroid, no? So this is the center point of our selection. And kung center point lang siya, di ba square yun or rectangle, siya lang talaga yung X and Y coordinates dun sa gitna. Dito naman sa center of mass, this is actually the brightness weighted average of the X and Y coordinates of all pixels in the image selection. So for example, if your data, kung sana wala yung kamay na naghulog ng bola, pwede nating gamitin tong center of mass. Kasi parang yung ImageJ, ang titingnan kung saan ba kaya yung center of mass based dun sa dun sa square natin, titingnan niya yung brightness nung ating bola, no? So siya yung mag-i-infer kung saan yung center of mass ng ating object. But in our case, I will use lang muna this Y kasi meron tayong first few slides, di ba, na nandun yung kamay. So, kung may bola, may kamay, makikita niya bright din yung kamay. So, hindi niya mai-infer yung correct din na center of mass. So, I will use this muna. And after this one, I will copy this into another sheet, and then I will create a graph. Ito na yung isa sa ating objective, to create a position versus time graph. Now, if you see here, kasi millimeters at first, di ba, yung Y natin. So, kinonvert ko lang into meters, kasi naka-meters yung ating gravitational acceleration. And then, I computed the delta T. Of course, this is 60 frames per second kasi ako. So, every delta T ko is fixed at 1/60. And then, my delta Y, and then my velocity. Now, from the position versus time graph, so Y lang to siya na position ang ginamit natin, no? Although you will see na medyo may konting X, pero since we are only looking at the Y motion, pwede nating kunin lang, i-separate yung Y. And you will see that the equation of the best fit line is a parabola. So this is the position is a function of the square of your time. And we know this to be true. Expected to siya sa ating free fall experiment. And if we plot our velocity versus time, so this time naman straight line siya. So you will see this one, and you get this equation of a straight line with a negative slope. And if we set our y-intercept to 0, in this case, pinilit natin siya to be 0 since alam natin that this should be 0. So ito yung lalabas na equation. Wala na siyang y-intercept dito. And you will see that the r squared for this one is 0.9997, which indicates a very, very high correspondence to a line. That means yung ating experiment, talagang ganito yung kanyang equation. No? So straight line talaga siya. And para naman dun sa second ball, ito naman yung lumabas, still, ano din siya, parabolic din natin ang ating equation. And for the velocity, that would be a straight line. Okay? So here, you will see from the free fall experiment that our velocity from the kinematic equations, ito yung pinakamadaling gamitin, the velocity versus y graph. And you will see that the minus g is actually our negative slope. So we will now have our experimental value for our gravity. And in this case, ito siya, minus 10 and 9.81 yung accepted value. So that's about a 2.45% error. So sa kabila naman, it's about 1.59% error. So actually, itong error margins na ganito, maganda na siya, di ba? Kasi madalas pag sa lab, when we do the actual experiment, malaki talaga yung error. In our experience, more than 10% talaga error ng students. Very fluctuating yung values. Kasi mahirap isabay yung pag-click. So merong delay. So this is a good way to appreciate the gravitational acceleration. Kasi ma-determine mo siya up to a value na maliit lang yung error. Okay? So let me move to my next presentation. So hinati ko to siya in two slides. So productive strategy. So now the second question is, what if the video recorded motion has a duration of several minutes? Okay? So what if hindi lang siya 20, like katulad ng slide natin kanina, di ba? 20 lang siya. So in other words, what if the image sequence consists of hundreds or thousands of images? So that's where TrackMate comes in, no? So TrackMate is a software tool for automated and semi-automated particle tracking, and it offers a generic solution that works out of the box through a simple and sensible user interface. The segmentation, filtering, particle linking processes and results are visualized immediately in 2D or 3D, allowing to judge their efficiency and adjust their control parameters. So if you look at TrackMate, you will see that there's a lot of, if you look at Google Scholar, so maraming mga publications using TrackMate. No? So some publications here are very interesting. For example, TrackMate is used in quantitating the cell, no? So turning images into numbers. Or here, TrackMate is used to track the cancer cell invasion in a brain microenvironment. So in this case, zebrafish na mga brain. No? So those are just some of the examples of how TrackMate is used. Ito naman siya. So you have 4D live imaging of and analysis of chick embryo. So you will see that the automated particle tracking using TrackMate is actually being used in legitimate talaga ng mga studies, no, in researches for publications. So let's try to do this here, no, very quickly. So pag Fiji yung dinownload nyo, yung TrackMate plugin, nandun na siya. So you just have to click plugins and then browse sa baba, and then you'll have your TrackMate. So very naman siya to use, very parang user-friendly. You can just experiment on it and then click back if you think you need to change the settings. So here, the default nung sinuggest niya is 10mm na blob size, and you will see na yung 10mm pala, hindi pala siya maganda, maliit masyado for the balls. So pwede mong palitan, pwede mo ring palitan yung threshold para makita niya ano yung background, ano yung hindi. And now you will see here, na pati itong scale sa gilid, inisip niya that these are also particles, di ba? So, kailangan mo munang tanggalin yan, i-crop out natin dun sa ating screen para yung makita niya lang as bola is yung ating experiment na mismo. So, crop natin siya, and then we zoom it in, and then we go back to our TrackMate. So, refresh. So actually, when you try this at home, no, so say, I think recorded naman yung video, and then you try this for yourself, you will see that very very user friendly naman yung ImageJ na TrackMate plugin. And yung guide niya on how to do it is also easy. So here you will see na nakuha niya yung mga spots at a very short period of time. So 20 balls. So 6 seconds. And then pwede na nating i-display yung track. So actually, more on next lang naman. Yung mga default niya na setting is already good. So click on next. And then ililink niya na ngayon yung mga spots sa bawat slide para makita niya kung ano yung nag-connect ng mga points, no? So dito nakita natin ay hindi niya makuha dito, hindi niya na-connect. So nilakihan ko into 70 mm na distance. So nakuha niya na yung motion ng bola. Pero of course meron pang mga mali. Ang nakagandahan naman sa TrackMate, we can edit the spots, no? So we can launch this track scheme and then we can look into the tracks na hindi tama. Dito, kasi meron tayong mga kamay, inisip niya na yung mga fingers are actually part of our tracks. So we can just select that and then delete that. Kasi we know na hindi yan siya kasama dun sa bola. So dalawa lang yung iiwan natin ng mga tracks. Now, another good thing about TrackMate is that you can actually edit the position of the spots. No? So you can move the spots, pwede mo siyang i-click, pwede mo siyang i-drag. Kasi at first, syempre, naguluhan si ImageJ kasi merong bright din dito na kamay. Unlike dun sa baba na black na lang, sakto na yung tracks niya. So you can actually edit that. Pag satisfied ka na, saka pwedeng saka mo i-okay, kumbaga. No? So edit mo lang, and then you do the same for the others. So pwede mong paliitin. So click ka ng next. So say ganyan, na-edit mo na lahat. At gusto mo na siya. So now you can visualize your tracks. No? So move ko na lang dito sa baba para mabilis na siya. So ayan, so pag okay ka na dun sa tracks mo, you can now launch itong analysis na part. So pag clinic mo yan siya mamaya, lalabas na ngayon yung file, yung table nung lahat ng mga lahat ng mga spots, kumbaga. So you will see the links. Yung kailangan natin is this spots in track statistics. Nandun na yung position ng X and Y natin. Yun ang gagamitin natin para naman dun sa ating similar na data, na table. Okay? So save lang natin ulit. Now you might think, bakit naman ako magta-TrackMate pa? Naman, parang napakadali lang naman yung 20 images, di ba? Magli-learn ka pa ng plugin. Pero what if dun sa experiment mo later on, like sa bio, nag-observe ka ng motion ng bacteria, tapos ilang minutes ang haba, di ba? So, kailangan mo ngayon ng mga ganong tools. Like in this example, 1,000 tong images ko. So, ang hirap, tas 10 yung aking 10 yung aking particles. Ang hirap naman kung ima-manual ko. So, this is how these are actually particles that are placed on a vibrated surface. So, mukha din siyang nagmi-mimic din siya ng motion ng certain organisms, no. If you're given this type of experiment, ang hirap na gawing mano-mano yung pag-analyze. But if you use TrackMate, so dito siya. So the tracking, you will see here that found 10,006 spots. So 10 lang yun, 1,000, so dapat 10,000 lang. So that means merong 6 spots na kailangan mong tingnan at i-edit. However, you will see that the detection is done in 130.5 seconds. So you will see na nakakatulong siya, no? 1,000 images in just 100 plus seconds. So ito na ngayon yung itsura ng mga tracks na ginawa niya. And you can click here kung gusto mo ma-visualize lang yung local tracks. So pwede mo ngayong makita yung motion ng mga particles. Isipin mo kung ikaw mismo yung naglagay ng mga mano-mano ng mga spots niyan, matagal, di ba? Mabagal. But here, because of TrackMate, you can do this semi-automatically. No? So you can edit also the tracks to make them perfect before exporting the final position. So yun ang advantage ng TrackMate. Now, if you want to learn more about TrackMate, so you can actually visit TrackMate dun sa kanilang website. And then they also have this paper in methods, no? So open and extensible platform for single particle tracking. And also, I also have a paper wherein I use my the TrackMate software or the TrackMate plugin in my publication dun sa ginawa ko dun sa dissertation. So, this is the itsura ng mga particles ko at that time. And ginamit ko yung TrackMate to track these particles. So, these are sample trajectories the results of TrackMate. So, you can also use this one in your experiments, especially if you're looking at motion sa biology or ano pa bang mga areas na merong motion. Kahit hindi sa biology, if you're tracking diffusion, for example. Sa chemistry, pwede rin siya. So these are some examples of projects ng mga students ko before last year. So ito siya for a single pendulum na merong dalawang pegs. So this is by Brian. And ito yung kanyang experiment. And this is the result sa TrackMate plugin. So tinrack niya yung pag-move ng pendulum. Ito naman siya is the result of Mark. So ito naman is a pendulum naman na merong spring pala. So spring massless system. So ito yung itsura nung kanyang motion as in TrackMate. This one is for a double pendulum. So ito yung para sa taas na mass, ito yung para sa baba na mass. And you will see that if you use these tools, you will get to appreciate more the system. Makikita mo din yung mga chaotic part ng motion, so you can explore the system deeper. So the TrackMate can be used in simple experiments and you can extend it to more complex experiments. Motion behind, and I think we only have a few minutes to spare. So I'll just go directly to this automated particle analysis of non-homogeneous granular mixtures. So here we want to count the number of particles in non-homogeneous granular mixtures and then visualize the size distribution of constituent particles using a histogram. So here, if you Google again, you will see that there's a lot of studies na kailangan ng particle analysis. Marami sa bio, sa chem, material science. These are other examples. So for example, you have this set of stones. Gusto mo malaman yung area, gaano ba kalalaki yung mga stones, Usually, surface area, projection nila on a surface. So, what to do? You can actually use TrackMate to do this thing automatically, Hindi na natin kailangan mano-mano i-measure yung size ng bawat stone. Pwede natin gamitin yung ating particle analysis. Usually, ginagawa ko, nagduduplicate muna ako ng image para hindi masira yung dati, So, click natin siya to be 8-bit. Click threshold. Kailangan naka-red lang yung part na gusto nating i-measure. Pag satisfied na tayo, nakita nyo naging binary siya na image. Parang light and dark na lang siya, So you can process it, you can click close or fill holes. Say pag mga butas-butas siya, pwede siyang i-close. We want the area. Yung area talaga ang pinakagusto natin kasi gusto natin makita ano ba yung size distribution. So we can click analyze particles. And marami din siyang options. Pwedeng outline, pwedeng overlay mask. For example, gusto natin ng mask. So, ito na siya. So, yun na yung ating stones. Automatically, nalaman niya na 20 pala to, lahat ng stones. And then, you will also see na nilabel niya yung bawat stone. So, say for example, imagine mo, pwede din tong bacterial culture, for example. Malalaman mo kung gaano yung surface area, gaano kalaki yung surface area ng bawat culture kasi merong label at meron ditong data sa kabila. You could also play around with the other masks, for example. So you can click on, say, outlines. Mas maganda pag outlines, mas makikita mo yung mga label ng numbers, for example. So you can play around the different options in visualizing. So yan, kita natin siya. So you can browse. So ImageJ automatically does this for you, and you will get a view of how the areas are distributed. So we can click on distribution and then area. So automatic binning, for example. So gusto nating malaman ano yung distribution ng area. So yan yung ating histogram. So you can also export the data from the histogram by clicking the list. And then you can then file and then save as. So ang ibig sabihin lang ng histogram na to, is that meron tayong 9 na stones na yung kanyang size is between 0 to 12 millimeters. Tapos merong 6 na 12 to 16, merong 5 na 36 to 60. So this this plugin in ImageJ gives you a tool to automatically do these things, no? Para makuha mo yung mga sizes ng mga gusto mong i-measure. So this is another example. This is rice naman. So again, we just crop this one. Tapos, so hindi necessary na black particle on white na background. Pwede rin namang white particle on dark background. That's also okay. No? So, process natin siya. Tapos,
Circle
so, ginawa natin
Wednesday.
siyang grayscale. Tapos, adjust, threshold again. Kita nyo, yung red, ang ni-red niya is yung background. So, ayaw natin nun kasi gusto natin i-measure yung bigas. So, ilipat mo lang dito sa slider. So, pag sakto na, pag gusto mo na yung red na pinili niya, saka mo i-click ng OK. So, you can manipulate. Otherwise, kung ito yung pinili natin kanina, yung background, yung kukuha niya ng area. So, ayan na. So, naging ano na siya. Click binary and then fill holes. So, close. Tapos, analyze and then click set measurements. Area pa rin ang gusto natin. So, pwede na natin i-OK. Tapos, analyze particles. So, marami tayong options again. So, parang katulad lang din kanina. So, this time, 50 siya. So, again, kung marami kang particles, mas maganda, no? Mas automatic. Now, you will see na meron siyang mga labels. But I will move on to the next na sample to show you na what if meron mga magkakatabi-tabi, no? Kasi iisipin siguro, paano pag merong magkakatabi? Well, ImageJ can still get that. However, sometimes you need your manual intervention, no, in this case. So say for example, meron kang katabi, so na-process mo na siya. So i-speed up ko na lang dito. Anyway, I will give you this video later on para pwede niyo talaga siyang, kasi mukhang mabilis ginagawa, di ba? Baka nahihilo na kayo, no? But I will give this to Joyce later on so you can play this, no, at your own speed later on. So suppose you have this one. It says 528. Now imagine if you have a bacterial culture, ganyan kadami, no? So you would appreciate this kind of tool. 528, malalaman niya agad yung area ng bawat particles mo. So a good advantage. However, you will see here, pag i-zoom mo, merong magkakadikit-dikit. Kasi white kasi yun, di ba? Hindi nakita yung edges, yung borders. But you can remedy these parts. Pwede mo kasing mag-manually correct, no? So, you can click this. So, you can draw. Dito sa ano mo, you can draw. You can zoom this in and you can draw with a brush, no? In my case, gumamit ako ng makapal na brush lang just to show. So later on, pag natapos mo na yan, say, correct mo yung mga edges, determine mo siya. So, pag i-click mo na siya ngayon ng analyze sa ImageJ, pag inulit mo, makikita mo na nahiwalay na yung mga dito na parts. No? Kanina kasi kinuha niya as one image lang. But now, pag i-zoom in mo siya, ano na siya? Ayan. Consider na siya as separate particles. So, yun ang gamit ng particle analysis na plugin sa ImageJ. And the applications, again, are endless. Hindi lang naman necessary sa granular matter, but in many fields of research. So, let's move on to the last experiment, contact angle measurements, hydrophobic versus hydrophilic surfaces. So, dito, we want to determine the degree of hydrophobicity of different sample surfaces using static contact angle measurements. Characterize the effect of increasing drop size on the static contact angle of water drops in different surfaces. So again, if you look at Google Scholar, you will see that there are a lot of experiments using contact angles, especially in material science or in vacuum physics, for example. So these are additional examples. So in ImageJ, merong mga tools din for measuring the contact angle. So when you have a liquid drop and you place it on a solid surface, it will spread to the surface, or pwede rin naman siyang para lang siyang magbilog siya, di ba? It will spread to a limited extent. Now, the wettability of the solid surface or the wetting ability of the liquid on the solid, it's determined by the final condition of the applied liquid on the surface. So if you have a surface, meron kasing binabalanse na interfacial tensions, no? Between solid and liquid, liquid and gas, and then gas and liquid. So ito yung ating Yung contact angle, yan yung angle dito na i-measure natin later on. So, alam natin na pwedeng spreading, pwede rin naman na talagang non-wetting, no? Yung ating material. Pwedeng negligible wetting. Pag equal naman yung ating tension ng solid liquid and solid gas, ganito lang yung itsura ng ating liquid. Now, ito yung mga types of surfaces at yung kanilang mga usual na contact angles. And ngayon, merong interest in superhydrophobic particles, especially or superhydrophobic surfaces, especially in nanomaterials. Meron din namang mas gusto nila na mas maging wettable yung surface. So, a lot of applications in both parts of the spectrum. So, this is an example of the drops that I've taken before in my master's para sa IIT. So, ito yung mga drops na napicturean ko. So, these are actually leaves of Aberoa bilimbi. So parang iba. Hindi ko alam sa Tagalog kung ano siya. So iba siya sa amin. So yung lotus kasi yung gabi-gabi na uso ngayon, yun yung popular, di ba, na super hydrophobic. But this one is also gearing towards super hydrophobic. So meron tayong tool actually na pwedeng gamitin, no? So in this case, I wanted to check muna the size. So that's why I used the rectangle, tapos i-click ko yung sizes muna. Kasi gusto kong makita ano ba yung effect nung paglaki ng droplets. So nagdagdag ka ng maraming drops, say for example, in your surface. Ano ba yung effect niya sa contact angle? So maiisip natin, dahil bibigat yung surface, so mag-change yung gravity, so malamang maano siya, no? Kung at first, parang nakapatong lang talaga siya sa surface, later on, it could be na mas didikit siya. Pag dinamihan mo pa ng dinamihan yung tubig, syempre mag-spread na siya after a while. So parang yan yung gusto nating makita sa experiment na to. So we just measure dito yung mga widths nila, yung sizes, so bounding rectangles. So after doing that, so ayan, na-measure ko na lahat yung mga widths. So meron na akong table. Okay? So isa-save ko ngayon yung table ng mga sizes na width tsaka height ng aking mga drops. So now I can do the contact angle analysis. So yung ginagamit ko sa ImageJ is yung drop snake na plugin. Marami pang ibang plugins actually. Ito lang yung nagamit ko na before several times. So madali lang siya, click on plugin, tapos drop analysis, drop snake. So using the cursor, magdo-draw ka lang nung bilog. Okay? So ito pala siya ng mga plugin. Originally, wala to sa ImageJ. Pero merong mga researcher na ginamit nila sa kanilang study. So sila mismo gumawa ng plugin tapos pinublish nila. You could actually do the same in the future, no? Pwede kayong magdagdag ng mga plugins sa ImageJ and then publish your own parang add-ons. And people will use them in their experiments. So you will see dito, pag nagawa niyo na yung automatically ilalagay niya dito yung iyong contact angle sa left and sa right. And you could also edit this one. So the good thing is, pag first mong bilog, hindi naman siya ganun ka-perfect, okay lang. Kasi pwede mo naman siya later on na galaw-galawin. So you don't have to worry na super perfect siya at first. So pwede mo siyang i-move around until you are satisfied with the contact angle. So that is how to use simply yung drop snake plugin. So ito na yung summary ko. So I checked the contact angles for the different drop sizes. So pareho lang to sila na ito yung kanilang scale, palaki ng palaki. So dinagdagan ko ng tubig hanggang dumami. So you will see that there is a slight decrease in the contact angle habang bumibigat na yung aking tubig. So essentially yun yung essence ng contact angle measurements. Ito naman siya ay sample mga droplets taken by a smartphone camera lang. So ito siya ay sa ating payong. So this is a fibrella na umbrella. So just to show yung itsura ng droplets on an umbrella. So these are just some materials. If you look at applications of hydrophobicity or hydrophilicity, so sa biomedical applications, they are more on hydrophilic coatings. Meron din ito siya fabrication of artificial superhydrophobic na lotus leaf-like bamboo surfaces. So meron namang artificial materials to mimic your natural surfaces. Ito naman siya, tinignan niya kung ano yung effect ng hydrophobic leaf surfaces, anong effect ng pesticide concentrate dun sa hydrophobicity ng leaf surfaces. So again here, the applications are endless with a simple tool, no? So ImageJ is not the end in itself. It will just be a tool in your experiment and in your future in your research activity. So even though you're at home, like say you can explore yung mga tanim-tanim nyo sa bahay, and then meron palang ibig sabihin yung hydrophobicity ng kanilang leaf surfaces. So in summary, sorry, mabilis masyado ako magsalita. No, but you can view this video later on or in the PowerPoint as well. I will give this to you. So in summary, these are the 3 experiments that we have looked into, and I hope you were able to see at bird's eye view lang or a grasp some techniques, and hopefully you will see that ImageJ is a very, very powerful tool. Although open source siya, it's or I mean, yun ang nakaganda sa kanya. It's open source. You don't have to buy it, but the power is very, very endless, no? Kasi pwede pa kayong magdagdag. So technically limitless ang applications in the future. So I think that's the end of my talk, and thank you very much for your time. Sorry for the very fast-paced na talking. So thank you very much for your time. So I will end my share screen, and I will give this back to our moderators.
Hey, thank you very much, Dr. Dengal. To our audience who just joined us, I am Al Vincula, a faculty from Mathematics Department, and I will be the moderator for the Q&A. So as I have read the questions from the chat box, I was really overwhelmed because we have a lot of questions, Dr.
Yeah.
Dengal. Okay. So let's answer as many questions as we can. From Cindy Hanelet Cascaro, halos similar rin po siya sa OpenCV. I think OpenCV means Open Source Computer Vision Library.
Personally, I haven't used OpenCV, so hindi pa, I cannot comment on that, but I can look into it later on. Kasi parang masyado na akong na-in-love sa ImageJ, so hindi na ako masyado naka-explore ng other say image processing software, no. And since nakikita ko sa mga publications din na ginagamit yung ImageJ, so parang nag-stick na ako doon. So wala akong ma-comment kung similar ba talaga siya sa OpenCV. So wala pa akong experience.
Thank you. From Alwilan Belio, what is the big advantage of using ImageJ compared to Tracker?
Hindi din ako nakagamit specifically ng Tracker software, pero yung ImageJ na TrackMate plugin, so far, hindi ko alam kung sa Tracker ganun din pero na-visualize ko kasi yung tracks and I'm able to edit the tracks later on. So, kung meron siyang maling tracks, I could edit it. At since hindi pa ako nakagamit specifically nung Tracker lang, so hindi ako sure kung ganun din sa Tracker. I'm sure marami ding ibang pang-track kung ang particle tracking lang talaga na capability ng ImageJ ang pagbabase. Marami pang ibang softwares din for tracking. So, the TrackMate plugin is, I think, fairly recent addition to ImageJ. So it's also a product of another research, no, ng somebody from the US. So parang 2017, 2018 siya nadagdag. So since gumawa ko ng thesis ng mga years na yun ang nagamit ko. But I think there are other optional trackers as well. So hindi ako masyadong familiar sa tracker specifically.
So a question from my student, from Miss Princess Katindig. Di po ba ma-affect yung dimensions or yung computation dahil po ina-adjust yung rectangle per frame?
Dun sa pag natin ng free fall motion, kasi yung tinitingnan natin is hindi yung size ng ating ating bola, but just the center of mass. So if we adjust the rectangle to fit doon, kasi di ba parang nagiging rectangle yung ating bola dahil nagbe-blur, no? So yung tinitingnan lang natin actually is the center. And although we can manually determine by eye kung saan yung center, instead of parang manually pointing to the center and see kung ano yung coordinates nun, we use the rectangle para siya na yung mag-average kung saan dun yung center of mass ng rectangle. Hindi naman siya affected. Like dun sa pinakita ko na na yung parang mga vibrated particles, center of mass lang din ang ginagamit nun. So usually sa mga tracking, you're looking at the center of mass ng motion. Unless of course, yung research mo is kinoconsider mo na yung shape ng material. In our experiment, okay lang siya since yung position lang naman ng center ang tinitingnan natin.
So from Phoebe Ambalada, so mas accurate po pag ImageJ kaysa sa manual?
If manual ba na experiment is yung manual talaga na free fall experiment, I would say in this case, yes, kasi so far sa mga students namin, yun ang problem nila eh. Hindi talaga malapit sa 9.8 meters per second ang nakukuha nila kasi nahuhuli sila sa pag-time. Pero if you have yung sophisticated equipment, like yung pinakita ko sa Pasco, very ano din yun, precise din yun kasi naka-time din yung switch nun. So, depende lang din sa equipment na gamit mo. In this case, if I compare it to the manual na ikaw yung magta-time ng motion, the video image processing would be more accurate.
Okay, thank you. From Joshelle Andrea Bechil, I think she's also a student. How can we ensure the reliability of results generated from the ImageJ software?
Well, for one, you can compare, say, if you are looking at to measure certain constants, for example, like what we did, the 9.8 for the gravitational acceleration. So you can compare it to your table value. And normally, if you are within the range 10%, acceptable yun sa publications, no, the 10% error or below, no, the closer you are to the value. And another way to do this is you can repeat the same experiment. So say, kasi kanina isa lang pinakita natin, di ba? So you can perform several trials. So say, pag ipapublish mo yung research mo, syempre hindi lang naman one trial gagawin mo. So you will say 10 trials, and then you will use, you will drop the ball several times, and then you will get you will get your G value. Marami ka ng G value, titinan mo ngayon kung magkakalayo ba or magkakalapit yung makukuha mo. Pag magkakalayo sila, hindi masyadong malaki yung variability. Like you can use your t-test, for example, no, to see kung significant ba yung pag up and down, for example. And makikita mo kung malapit lang talaga siya dun sa value. So, you perform several trials and that's how you can see na, okay talaga yung nakukuha mo. Kasi pagka sa next, malaki na yung hindi siya reliable. However, like when you publish your research, it would help to study kung sino mismo yung gumawa ng specific plugin. For example, sa TrackMate, meron talaga silang published din na PDF on how to use the TrackMate plugin. So you can then cite that and parang you will trust them that this is reliable because they have already published this method as well. Parang ito, gagamitin mo na lang siya na tool for your own study. So yun yung mga ways I think to verify.
Okay. So the next question was already addressed about reliability of the ImageJ. From Sally May Maranan, maapektuhan po ba yung result if yung ginamit po ba natin way of converting that MOV to ImageJ is yung convert to .avi instead of convert to image sequence?
Yes, by experience, it could be affected depending on the video conversion that you use. Karamihan kasi nung mga pang-convert na mga free from .mov to .avi, papansin nyo, i-change niya kasi yung yung pixels, yung parang minsan magiging ganon, makukompress siya or magaganon siya. So, change yung kanyang pixel size. So, I think na-experience ito ni Joyce in converting the .avi videos. And personally, na-experience ko. So, minsan, nababago yung dimension. So, once nabago na yung dimension, mag-iiba na yung mo. Although you still have your scale, di ba? So, you can base the scale kasi pati yung scale mo sa gilid, nagbago din yun ng dimension. Pero, yun ang isa sa reason talaga, kaya gusto ko yung convert to image sequence kasi kung ano talaga yung original number of pixels mo, yun din ang kanyang output dun sa image sequence. But if your converter from .mov to AVI does not change the dimensions, there's no problem in doing that route. That would also be good. Just make sure na hindi na bago yung number of yung size ng pixels ng video mo.
Okay, thank you. So I have a question from my student. I think I will answer this in our class. I'm just curious, how do you create the graph in Excel?
Yeah, you can do that.
Yes.
Yeah.
So about creation of graph in Excel. So I can teach you that. There's a question from Cyrus Itang. May instances po ba na mas okay gamitin ang iba kaysa sa ImageJ?
Dun sa pag-track kasi so far sa ImageJ ako nagta-track So manual and I would say reliable naman siya since tinanggap siya, since na-publish siya, so tinanggap siya. So maybe the reviewers think that the method that I used was legit naman siya. As opposed to well, when it comes to image processing na yung image mo medyo dark, parang minsan like your video is not that optimal. Yung pag more on image enhancement, mas gumagamit ako ng Photoshop for that. So minsan, pag meron akong video, tapos ginawa ko siyang image sequence, tapos when I try to use ImageJ, hindi maganda yung kanyang you would have to do pre-processing kumbaga. So you can use GIMP siguro, or in my case, I use Photoshop, ganun, to pre-process the images before loading them to ImageJ. So hindi naman talaga sa lahat ng bagay, ImageJ ang pinakamaganda. So you have to have a lot of skills in various softwares din.
Okay. So I think we can entertain 2 more questions because I have here 5 pages question, Dr. Dengal.
Well, you can send them to me and I
Yes,
will
from
try
the
to
chat,
answer
yes.
them, no, via email.
Yes, I'll do.
Yeah.
I'll do. Thank you. Okay, from Fatima Lakadin, compared to manual, would you say that the results that the ImageJ gives are more precise?
I would say not necessarily, not always. Say for example, yung paggawa ng mga bilog, because depende yan sa tao. Kasi if you're very if you want to be very precise, pwede ka pa ring makakuha ng precise na ng manual mo, no? Kasi sa ImageJ, syempre mga yun eh. Ginagawa nila yung parang machine na AI lang, 'di ba? Titingnan niya saan kaya yung bilog na Kaya meron talaga siyang option for the person doing the analysis to correct it, no, to manually annotate, no. I think the pinaka-advantage sa ImageJ is the automation when you have thousands of images. Like in my study, 10,000 images per trial ako. So I had to divide it into 1,000 images per batch kasi hindi din siya kaya sa computer ng 10,000 images at the same time. But the good thing is, after siya na-automate, hindi ko iti-take agad yung results na yun, no, ng tracker. I have to click one by one to see kung sakto ba talaga yung positioning. And then, kung merong lalo na pag magkakadikit yung mga particles, magkakaroon siya ng na malilito siya kung saan yung nag-move into which, then you can do the manual annotation. So, hindi mo siya itatrust lang talaga on its own, but it will help you to speed your process as compared to doing it manually. Pero pag like yung 20 images lang, I would say the manual tracking or manual measurements in ImageJ would be good. But if you go to hundreds and thousands, then you would do the automated thing. But still, you need the human intervention for your results to be precise.
Okay, so our last question for this Q&A portion from Ms. Janet and May Pagaw. What are the possible sources of error po na madalas nangyayari sa TrackMate and dapat iwasan in order for us to attain accurate results po?
Sa TrackMate, by experience, pag nagkakadikit yung particles, so hindi ko din siya masasabi na pwede siyang iwasan since pwede rin naman ma-improve, no? So when you are experimenting kumbaga dun sa dissertation ko, di ba meron akong mga particles na nagmo-move together. So yung TrackMate kasi, i-guess niya kung sino yung, say, dalawang particles, tapos nagkabit, tapos gaganon. Minsan, kasi computer lang naman siya, no? So hindi niya alam, maguguluhan siya kung ito ba yung gumanon, or nag-cross. So pwede siyang magkamali sa pag-determine ng mga ganong klaseng scenarios. So maganda, in your experiment setup, well-lighted talaga siya. Mas maganda kung kasi kung mas may shadow, yung experiment mo, hindi siya well-lighted, so magdadagdag pa ngayon yung shadow, no? Another thing that you can do is make sure that the contrast of your particles and your container is also good. Parang dun sa experiment natin, dahil black yung background ko dun sa freefall, kita niya dun sa automatic na, ito yung bilog kasi lighter gray siya compared sa black. If you do your experiment na medyo colored yung background mo, tapos colored din yung bola mo, pag ginawa mong grayscale, medyo konti lang yung difference, mahihirapan si ImageJ. So those are the things that you can do to improve, no? At mas magkakaroon ng konting chances na magkakamali siya, no? So lighting, first and foremost, tapos pwede mong kulayan yung particles, so choose the higher contrast, and then I think yun lang din ang within your reach kasi the rest ng the motion hindi naman natin mapipigilan, say kung ano yung motion ng something, no? So yeah, I think that's it.
Okay, So thank you very much, Dr. Merchbel Dengal.
Dami pala talagang questions.
Yes,
You can
marami.
you can
We
just
have 5
email this
pages.
to me and I can reply to them no para naman masagot natin yung mga concerns ng iba.
Yes, yes, Dr. Dengal, we will do. Thank you, thank you. We wish to entertain all the questions from our very enthusiastic audience, but we will be lacking time in doing so. Nevertheless, rest assured that the rest of the questions will be sent to our speaker. And to our audience, kindly send me an email to a.advincula@feu.edu.ph to direct to you the answers to your posted questions. And now, I humbly request our audience to answer the survey from the link that I'm going to show you on screen. Okay, the survey can be found at https colon double slash bit.ly slash capital M small a k I n g capital S e n s e capital S and small u r v e y. So you can see that on your screen. And to request for your e-certificate, you can do so by filling out our form in http colon double slash bit.ly slash M or capital M small a k I n g capital S small e n s e capital E small c e r t. And I have also posted the complete link in the chat box. Okay, so thank you for that. And as we approach the end of this webinar, we are also asking our participants to open their respective cameras and wear their best smiles for the picture taking. Okay, thank you for your cooperation. So kindly open your cameras. Yan. So medyo mahaba pa to. So bear with me. We have 8 pages of pictures. Sige, 1, 2, 3, smile. Okay, another slide. 1, 2, 3, smile. Okay, another slide again. Third slide. We have 5 slides to go. Smile. Thank you. Fourth slide. Smile. Thank you. Then another fifth slide. Loading. Okay. Smile. Next slide. 2 to go. Smile again. Thank you. And okay. Smile. And last slide. Thank you. Thank you very much for your cooperation. So before we conclude this webinar, may we once again call our department chair, Professor Frederick Helia, for the token of appreciation for our speaker for today. Sir?
Yes. Kindly display the A certificate. Hello, sir.
Sir. Okay po. Wait lang, sir. Thank you. Okay, so here you go. screen and this one.
Okay, good afternoon ulit. Far Eastern University awards the Certificate of Appreciation to Merjibel D. Dengal, PhD, for her valuable contribution as speaker during the Physics Webinar Series 2020 on making sense of the physical world one image at a time using ImageJ in work from home science experiments. Given the 6th day of November 2020 at Far Eastern University, signed Frederick Helia, Chair, uh, Department of Mathematics, Dr. Rowena Capolong Reyes, PhD, Dean, Institute of Arts and Sciences. Marami po salamat po sa ating speaker.
Maraming salamat din po sa pag-invite. It's my honor to be with you all.
Sir Al.
Yes, sir. Stop sharing. Okay. Okay, so we have realized in today's presentation that ImageJ can be an indispensable tool in exploring laboratory activities and researches in physics and other science fields. So one most interesting fact that I think is worthy of sharing is that this tool is free and is available for everyone. So we would like to thank the presence of our teachers, educators, professors, and students from Department of Education, Philippine Science High School, Mindanao State University, Marawi Campus, Far Eastern University, UP Visayas, and uh, um, guests from other fields. So we would also like to thank Dr. Rowena Reyes Capulong, our Dean, Associate Dean Marquisla, for the poster, Sir Richmond Lim and Sir Christian Evasco, our experts from the technical fields. Kindly receive our sincerest gratitude, our dear audience. Thank you for your time and effort, and God bless us all. Thank you very much for attending. Thank you, thank you.
Maraming
Thank you very
salamat.
much, guys.
Thank you.
Maraming salamat sa inyong lahat.